All right. It is 12:30, so we'll get started for today. So first, some announcements. Test 4 next week, Tuesday. So test 4 is going to be like, or not test 4, test 3. Getting ahead of myself. Test 3 next week, Tuesday. So test 3 is going to be like the other two tests. Same kind of format, probably. I haven't written it yet, so I don't know. But most likely there'll be a lot of multiple choice there, some short answers. Nothing you haven't seen before in terms of how I do my tests online. This material that we talk about today and everything past this date will be on test 4. And test 4 will be given like in the last week of class. That way you have seen all the material before you get to the final exam, which is cumulative, right? So just be aware of those deadlines coming up. But other than that, we can get started. Oh, yeah, I forgot. So homework. Since I assign homework after every lecture, and it's due in a week, normally that's been okay for our test dates because the homework that was due on the test date directly related to the test. So it was an incentive for you to do that homework before you did the test. However, for today's lecture, since the material is not going to be on test 3, the homework that I will assign for this will be due on next week, Thursday. So the homework for this lecture will be due, what is that? 7 plus 6 is 13, the 15th. Let me pull up my calendar and make sure it is Thursday the 15th. Yep, it is Thursday the 15th. So you'll have 2 different parts, part 1 and part 2. So make sure you look out for that when you're looking at your homework on Wiley Plus. You won't have any homework due next week, Tuesday, but you will have 2 parts due on Thursday from this week. And while I'm on that, one more announcement, no live lecture on the 15th. I will be busy that day, and unfortunately I cannot be here for the 12:30 to 1:45 time slot on Thursday the 15th. I will have a recorded lecture that I'm planning to do on Wednesday or possibly even Tuesday of next week or on the weekend. Actually, I'll have a recorded lecture up, so watch that. It will be there, but don't come to the Zoom meeting at 12:30 Thursday the 15th. I won't be there, and I'll be sure to put up a reminder for that as well. Right, I think that's all the announcements. If I think of more, I'll say them during the lecture. But let's talk about DNA replication now. And we're going to start by talking about our model polymerase, DNA poly 1. DNA poly 1 is the first polymerase ever discovered in E. coli. And the person who discovered it won the Nobel Prize for isolating poly 1. Now, even though DNA poly 1 is our model polymerase, it's not the major polymerase in E. coli, but the mechanisms more or less work the same. And it is one of the most well-studied polymerases, and that's why we talk about DNA poly 1. But poly 1 in E. coli, it will synthesize the lagging strand and only a small portion of the lagging strand. Basically, all DNA poly 1 does is that it will remove the RNA primer of the Okazaki fragment. It will chop that up, and it will replace it with DNA. And DNA poly 1 is what we call a processive enzyme. And when we talk about polymerases, you hear the word processive a lot. Some polymerases are processive, some have very bad processivity. What a processive enzyme means is that you'll do multiple reactions without falling off the template. Different polymerases have different levels of processivity. DNA poly 1, DNA poly 3, which we'll talk about today, have very high processivity. A polymerase in our own body, polymerase eta, which is basically a polymerase that does last resort DNA repair. Like at your very last resort, the processivity of that polymerase is really, really bad because it makes mistakes all the time, but it is a last resort. So different polymerases have different processivity. When we're looking at DNA poly 1, it's kind of unique in that it contains two different exonucleases. It can cut either in the 3' to 5' direction or the 5' to 3' direction. Most polymerases don't have both of these functionalities. So DNA poly 1 is unique in that case. And it has these two different modes because it needs one mode to chop off the RNA fragments, right? That would be the 3' to 5' exonuclease. While it can also serve as a proofreader for itself. So let's say it put in the wrong base, it would need to go back and chop off that base. So that would be a 5' to 3' exonuclease activity. So two different exonucleases in the same enzyme, one for removing RNA fragments, the other for removing an incorrectly placed nucleotide. And with this exonuclease activity, DNA poly 1 is really good at its job, which you would hope, in that it only makes an error in 1 every million to 100 million bases. E. coli, I don't know the number off the top of my head, but it's only like 6 million base pairs, I want to say. I might be wrong, but basically it only makes an error once every few generations. Which is what you want. You don't actually want your DNA to mutate, even though these times it does make an error, those mutations can lead to a change, which is evolution. So errors happen very infrequently, but they do happen. And the process of removing the primer and to add DNA has a proper name that's called Nick translation. Basically, let me here. The gap between nucleotides, if you don't have a phosphate bond here, as listed here, that's a nick. So when you're missing a phosphate between two different nucleotides, that's called a nick. And so DNA poly 1 can fill in that nick, no problem, nick translation. So we'll just go and fill in the rest while chopping off RNA. So that's what we're going to be looking at to begin here. But I do have one question, even though I'm putting up more and more reading guides, worksheets. But here's a question that I thought out while making this stuff. It's been a while since we talked about the structure of DNA. So tell me, where could a 5' to 3' exonuclease activity could happen on that image, and also where could a 3' to 5'? So you don't actually have to tell me. See if you can just imagine where you would think a 5' to 3' cut would happen, and where a 3' to 5' cut would happen. See if you remember your sugars. So I'll give you a just a minute to remind yourself of how you number sugars. All right. So remember, when you're numbering sugars, carbon 1 is going to be right here, like DNA, RNA, 2, 3, 4, 5. When they're connected to a nucleotide, that's why they get these prime numbers. So this is 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. So that's the numbering of our carbons. And if we're cutting 5 prime to 3 prime, that means we're going to be cutting right here. And, oh, sorry, I'm doing it backwards. I did 3 prime to 5 prime. Let me do, this will be in black. And this will be in red. All right. So if we're cutting 5 prime to 3 prime, let me do all my numberings here. 2, 3, 4, 5. 1, 2, 3, 4, 5. So here's a 5 prime to 3 prime. You would cut right there. 5 prime to 3 prime, you would cut right there. That's what a 5 prime to 3 prime cut would look like. 3 prime to 5 prime, that's the one I was drawing to begin with. 3 prime to 5 prime, 3 prime to 5 prime. So the cuts are slightly different, but you're basically cutting the same phosphate, and it's really what direction are you reading, right? 3 prime to 5 prime is reading this direction. 5 prime to 3 prime is reading the reverse direction, right? That's why that 5 prime to 3 primes, this is kind of like that proofreading, because you kind of see what you added, and it's like, oh, this is wrong. Let me cut it and go back and try again. So questions about numbering our sugars, exonuclease activities, or anything brought up so far? Right. So let's continue on with the main part of polymerase 1 that does the majority of the synthesis, and that's called the Klenow or Klenow fragment. So let's look at this picture down here. Here we have complete polymerase 1 on the right. And what you can do is you can chop this polymerase 1 into 2 different parts, the Klenow fragment and then a separate smaller domain. And we're interested in the Klenow fragment because this appears in all polymerases for the most part, and it's doing the addition of our nucleotides, right? So if we look at the fragment, it kind of looks like a hand. If you're like curling your hand and like kind of like cupping it, right? That's why it's thumb, fingers, palm. And that's, like I said, that's the majority of where the reaction is going to happen. Roughly 600 residues, 324 to 928, has our 3' to 5' exonuclease. And it's also where we're going to be adding our new nucleotides, which we're going to go over in a separate slide. The smaller domain is where your 5' to 3' exonuclease is, where we can start removing some primers. And when we look at this, how it binds, it's kind of hard to see, but there's actually DNA in this picture, right? There's your DNA. I guess it's also down here. So they try to outline it in the red. That's, you can see the double helix happening right there. And their different strands are purple and blue. So there's DNA actually binding into our fragment. And you're going to bind the phosphate backbones right in the cleft here. So right where my pen is, which makes sense that you would want to bind phosphates because DNA is meant out 4 bases, but the backbone's always the same: phosphate, 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 phosphate. Therefore, based on that knowledge, what type of amino acids would you expect to find in the cleft for polymerases? What class? if we're going to interact with phosphates. Going to have hydrophobic, hydrophilic, neutral, negative, positive. What type of amino acids are those like classes? Positive, that's correct. Yep. So maybe some arginines, but it's not going to be that. It's going to be lysine. No, that's not lysine. That is leucine. Lysine is going to be the main catalytic amino acid there. But yeah, phosphates are negative. Let me draw that phosphate for you. Very negatively charged. So to interact with that, we need a positive amino acid. So that's what we would expect when we looked at the cleft. When looking at this fragment, we see that it has the active site, and the active site is complementary to accept our Watson-Crick base pairs. That is, it is really made to allow A and T to be in there, or C and G. And how this really works is that for the base pairs, and they're trying to show it here, it's really messy, but they're trying to show with these spheres what's going on here, is that near where you're going to add a new nucleotide, you have base pairs that is forcing the DNA into the A conformation. The A conformation is not the normal conformation. The normal conformation is B DNA. This is quote unquote normal. That's how your DNA usually is. So when we're binding in our fragment, we are actually distorting the DNA a little bit. So it's in this A conformation. And when that happens, we're distorting the DNA so that the amino acids, that's what AA stands for, amino acids. The amino acids are hydrogen bonding with the bases. Now, if we're adding a new nucleotide and we do not form a Watson-Crick base pair, the hydrogen bond between the protein and the DNA gets disrupted. Why is that useful? Why is that a good thing? That when we form non-Watson-Crick base pairs, that the hydrogen bond between the bases in our protein are disrupted. Safety check, proofreading, exactly. That's how the enzyme knows if it's put in the right nucleotide or not, right? Remember, these proteins aren't smart. They have no intelligence. It's just, oh, if those hydrogen bonds are broken, let me bring in exonuclease and cut whatever's there because I must have made a mistake. So that's one way why it has such high fidelity, why it's so good at putting in the correct nucleotides, that those hydrogen bonds being disrupted is the safety mechanism that only fails 1 out of like every 1 million to 100 million times. So it's way more accurate than humans at making mistakes, I'll say that. 2 different forms, and they're called the open and closed forms. You can kind of see that with the distance between the finger and the thumb. Remember, the blue is the thumb. If you're looking at your hand right now and it's cupped, the blue part's the thumb, the green part is called the fingers. Right. And so if you, I don't know, if you look at the little image of me, I don't know if you can pull that up in screen as well, but this is basically what they're showing with the cup. So open, closed, open, close. It's just kind of like crushing on itself. And open allows new DNA to go in. Close, it's sealing that phosphate bond. Sorry, I didn't mean to clap if you weren't in headphones. I was probably loud. But that closing makes the new phosphate, and your DNTP, remember N stands for nucleotide, triphosphate, D for DNA. You'll lose pyrophosphate, 2 phosphates, or inorganic phosphate, and that's where the energy is coming to make this new DNA bond, this new phosphate bond. And that is basically how polymerases work. Open, close, checking to make sure that the hydrogen bonds are correct to see if the correct amino acid has been added. Different polymerases have slightly different fragments, like the one I was talking about, Paul-Eta, which is our last resort. It doesn't work. It's our last resort. I don't know what my template is. That fragment looks a lot different because it doesn't check for hydrogen bonds. It just puts in whatever residue it can find, basically. But for the most part, like the ones that are actually accurate are going to have that hydrogen bond check. So if we take a deeper dive into our Klenow fragment, so we're zooming in right at the site of catalysis, and this is what it looks like here, where here's our primer, right here. And here's our new nucleotide, our DDCTP. And you can see it's DD here. The reason why this is DD and not DCTP is because this was done for an experiment to try and capture the moment a new nucleotide comes in and then like kill the enzyme. So with DD, that means you're missing 2 hydroxyls, not just one. And so the polymerase can bind, and then it will try to add this nucleotide to the primer, but it can't because it's missing a whole hydroxyl, so it gets stuck there. Then we take a crystal, make that a crystal, and we shoot it with a laser, and you get this pretty image to figure out how this enzyme actually works. And what we see is that the active site usually has 2 metal ions. That's A and B here. The ions are usually magnesium, not always, but a good percentage of the time. And they're going to be next to 2 invariant aspartic acid side chains right there in the palm. Now, this is something we talked about in Biochem 1, so let's do a little review. What does an invariant amino acid mean again in terms of protein evolution? What does invariant mean? Can't be substituted. Yep. In every single polymerase that we find, aspartic acid has to be there. That means that it's critical for that enzyme to work, right? If you put in any other amino acid, that polymerase now dies. Then based on that image, why aspartic acid? I guess I'll answer this instead of having people type it up. This was an in-person class. I would call on one of you and you would talk to me, but unfortunately, we can't do that in this type of classroom. So you can see that aspartic acid, if you remember what aspartic acid looks like. That's an N. Let's see, double bond up. So that should be aspartic acid. And so we have 2 oxygens. Here they are. They are each of these oxygens are holding the magnesium in place. Here. And they're holding them in perfect place, perfect location, so that when a trinucleotide binds, they are warped into a position to interact with both of these magnesiums, weakening the phosphate. So it's going to be much easier for the PPI to leave because it's getting bent now. The bond is bending, helping to break that bond. But you're also positioning your phosphate that's going to be attacked and your primer is hydroxyl. So now they are in close contact with each other, and they're in the right orientation for that to happen, right? And then that's going to pop off. And that as well, what you're doing is these metals help to shield all that negative charge because you have aspartic acid, phosphates. And what does shielding mean again? What do we mean when we say like we shield a charge? Like charge balance, make it neutral essentially. Kind of, yeah. I think more of it like blindfolding all the charges, more or less. The way I think about it is you have negative charge next to each other. They hate each other. They repel each other. You put a positive charge in between them. I'm happy. I'm happy. I don't even know this other negative charge really exists anymore because I'm being shielded from it. But yeah, the charges are still there, but it's kind of like they've been neutralized more or less. It's just allowing like charges to come near each other as long as you have an unlike charge in between them. So that's not only is magnesium positioning everything for our catalysis, it's also shielding all of this negative charge to allow it to come together in the first place. So that's the inside look at how this enzyme works. So any questions about that information? All right. Now let's talk about how replication starts in the E. coli. Again, E. coli is our model system here. Eukaryotes start a little bit different, but we can get the basics of it. So replication initiation in E. coli. Hope you enjoy my awesome drawings here. So let's start. Replication in E. coli begins at a specific site called the ORI-C. ORI stands for origin. It's 245 base pairs in length. And before replication, it's in its double-stranded form. And the first molecule to bind is called DnaA. And DnaA binds to the ORI-C at an AT-rich region. That's roughly 45 base pairs, and starts to open this up by taking ATP and making it ADP. So it's using ATP to open up the ORI-C, and you have one DnaA, so this blue, bind to each side. Why does it make sense we start at a AT-rich region for replication? What's the logic of that? Or I guess another way to ask that question is, why don't we start at a GC-rich region? H bonds, H bonds, H bonds. Remember, if you don't know something in biochemistry, just say hydrogen bonds and you got a chance to get it right. Yep. GC, 3 H bonds between the paired. AT, 2 H bonds. takes less energy to melt open an AT-rich region than it does to open up a GC-rich region. So AT is our first region to open up. It's just way easier to do. So once our AT-rich region is opened up on our origin, the next two molecules, well, the next molecule to really bind is DnaB. Actually, two molecules are going to be involved here, DnaB, DnaC. So let's go over the pictures, this one from Google and this one my handiwork. So DnaB is going to be our helicase, and it's a hexamer. So that's why I have 6 little circles. It has 6 subunits. And it's going to bind to each side of our bubble that we're opening up. The way it binds is that DnaB comes along with a helper protein called DnaC, shown here. So this is, I'm just going to label my image here. And those labels go here. DnaC is what loads DnaB onto our bubble. And it's going to be B that unzips the DNA. Each DnaB will unzip 5' to 3', and you're just going to go in each direction, right? Because remember, E. coli is a circular genome. So basically, one replication is going to go this way. The other replication is going to go this way. And they're going to meet somewhere in the middle and hit each other. And that's when they know they're done. But that is the start of replication. Starts with A, then B, loaded on by C. Starts at the origin. And the first thing to unzip is the AT-rich region. Any questions about replication initiation in E. coli? And these pictures down here are actually what the molecules look like. 1, 2, 3. So this should be DnaB down here, I believe. But, I mean, I think I did a pretty good job too. That was a spark of inspiration. All right, now moving on. And let's actually look at how DnaB, which is a helicase, and remember helicases, they unzip DNA. Let's see how it works. And this is called a hand-over-hand mechanism. So imagine you're trying to climb that rope in gym. So if you're in school, so if you didn't have that at your school, what I'm talking about is a long rope that touches the ceiling. And one thing in gym class that they have you do is just go and climb to the ceiling, kids. That's safe. But when you do that, you do one hand over another hand over another hand, right? That's how DnaB moves along the DNA. So let's look at this picture. Here we're looking at double-stranded DNA. Well, this is one-stranded DNA, but it's going to be joined by the other strand here. This part in red is what's already bound to DnaB, our helicase. The green is where we're going. The different units, A, B, C, D, E, F, are the 6 hexamers of DnaB that we talked about. And when we're doing this, we're going to use nucleotide triphosphates. Here we have ATP as the energy to move our DnaB, but in reality, DnaB is one of those rare molecules that it can also use CTP, GTP, DTP, probably UTP. It can use any triphosphate as energy. It doesn't have to be ATP. And what happens is, let's say we're bound. So we're going to start a movement here. So we're going to chuck the, here in this case, the ADP from our last movement that we already used. When we remove that ADP, we have ATP being bound to our beta subunit. So ATP to ADP. That's what's going on here. We're going to use that movement, that phosphate, to move this subunit down 2 nucleotides. Well, it's actually 12 nucleotides, but it's 2 nucleotides below B. So 2 nucleotides here, 12 from where it was. And that separates DNA by 2 more nucleotides. And then you just repeat. F's the next one. So the ATP that was bound to F comes off, moves down, recruits a new ATP. And you just keep on moving and moving and moving. And when you're doing that, though, you're going to be building up strain, right? Because this thing is a circle. Actually, let me draw a little circle of DNA. Right, all of this is connected. And I really wish I had some rope on my connected to my wall right now, but when you are untying a rope that's hung up on itself, right, that's coiled on itself, when you start undoing the ends and pulling them apart, what happens is that further down your string, the tension will build up. It will just become tighter and tighter and tighter the more you undo the beginning of the string, because they're wound up on each other. Eventually in DNA, that will cause the DNA to break. The torsion will become so big, you'll start to break bonds. So there's a molecule called gyrase. What gyrase does is that it's further down from DnaB. So gyrase, so just example, if like DnaB is here and it's unzipping this, gyrase might be like here. It's going to be more pronounced than that, but that's the idea. It's just further down and we're going that way. That says gyrase. And what gyrase does is that it will cut one strand of DNA. And let me, sorry, I'm going to get off the PowerPoint here because I can show this better with my fingers, I think. All right. So so we imagine that we're in a double helix, right? Just double helix. What gyrase will do is it will cut one DNA strand and unwind it, because everything's wound very tight because of DnaB, unwinding it from the very, very start of our double helix. Gyrase will cut further down the line and like unloosen it a couple turns, then reattach, which relieves all that torsion. And then DnaB will continue to unwind it, building up torsion again. Gyrase moves further down the line, cuts DNA, unwinds it, reattaches that phosphate, and we continue on. So that's how we make sure that our DNA does not break due to torsional strain from unzipping. Yeah, any questions about the mechanism of DnaB or that information? I really got to remember to have string by my desk. Topoisomerase does that too, yes. Yeah, gyrase is so a topoisomerase is like the class. Gyrase is like the specific one in E. coli, but yes, humans have like topoisomerases that do the similar job. All right, so we're unwinding, and we're making single-stranded DNA. And DNA does not like being single-stranded. It wants a hydrogen bond. It will naturally want a hydrogen bond. So we need to prevent that. So to prevent our single-stranded DNA from reannealing to itself, we have single-stranded binding protein, SSB, which is I have shown here in these green spheres, also shown here. So one copy of SSB is shown in blue. One copy is shown in green. Right. So actually, sorry, I got that mixed up. The green and the yellow is the protein, and the blue and like the magenta here is the protein. So that's where your two copies are coming from. And what single-stranded binding protein does is that it absolutely coats the DNA. They bind end to end. That's what I'm trying to show here. Like there is no free DNA peeking. Like there should be another sphere right here. It binds incredibly tight, right? And then it will only bind to single-stranded DNA because the binding cleft is too narrow for double-stranded DNA to fit in there. So once single-stranded DNA is present, it just binds there incredibly fast and very tight to prevent that reannealing. And to facilitate that binding, we have a lot of positive charges. Again, makes perfect sense. The only thing that's similar about DNA is that it has a negatively charged phosphate backbone. So no matter what the base is, you're always going to have that phosphate backbone. So bind to that. During replication, we need to kick these off, or else we can't access the DNA. So that's the job of polymerases. When polymerases are actually going to read this DNA, they will remove SSB. But just remember our key, we haven't got to the polymerases yet. This is the yellow is DnaB, and that blue was DnaA. So DnaB is our helicase unzipping. And then a separate molecule, a polymerase, will go and chop these off. Well, not chop them off, but push them off. Just like a train moving through the tracks. Anything in its way just gets hit to the side. So let's actually get to the whole complex that is needed to replicate DNA in E. coli. And this is like much simpler than human replication machinery, which is why we study it first as a gateway in, and to try and learn the main concepts before you go into the much more complicated human machinery. So first things first, before we can add any DNA, we need a RNA there, an RNA primer. So the very first thing that happens is that we put an RNA primer. For the leading strand, we need exactly one primer because it's the leading strand. So once we have that primer down, we just continue till we're done. The lagging strand, you need many primers, which is shown here in green, to make our Okazaki fragments. The machinery that adds RNA primers is called the primosome, 600 kilodaltons. So it's a very big protein. And it's made out of a lot of different things. So it has our helicase with it, has the thing that actually synthesizes RNA called DnaG, and other subunits as well that we're not going to go into. But yeah, so the thing that actually is adding our primer is going to be our DnaG there, but that's the primosome. The whole purpose of the primosome is to add RNA primer. DnaB is what's going to be moving our primosome along, because we are going to continually need to add new Okazaki fragments. Right? So that's what's being shown here. So right now in this image, we are synthesizing DNA this direction, right? This is a lagging strand. So our polymerase right here is going to go and fill this in. And the next part of DNA is where the primosome is gonna start adding the primer. So the primosome is gonna move along here and maybe add a new primer here to create a new Okazaki fragment. DnaB, our hexamer helicase, that is continuing to unzip, going this direction. The primosome is going to follow it and put down new primers to continue with our Okazaki fragments. So that's the machinery of priming, and that's how we continually add RNA primers for our Okazaki fragments. The primosome, mainly due to DnaG and DnaB helicase. That's kind of what the images are showing here. New Okazaki fragment, old Okazaki fragment, new one starting. When you are synthesizing the actual DNA, in E. coli, we call it the trombone model. And I put a picture of a trombone there in case you can't picture one in your head. And it kind of looks like a trombone in that the DNA is kind of like coming back on itself, and then you have this long arm, and then you have the other part. So I can kind of see what they were going with there, especially in this picture, I guess. But when looking at the replisome, so the replisome is the machinery, is what we call all of the enzymes responsible for replicating DNA. That's a replisome. We have two copies of our polymerase. And our main polymerase here is DNA poly 3, which is shown in the blue. So copy 1, copy 2. So DNA polymerase 3 on our leading strand just continues on and on and on. It's fine. It doesn't need to fall off. It's going in the right direction. The lagging strand, though, it's interesting. One, since, we're always kind of like going back in time, and like, our new Okazaki fragment is going to start up here, but we have to finish the old one first. And this is all done by the same machinery. We have to make sure that the DNA we're synthesizing is connected to our replisome. Because and we're going to see this in the next image, these two polymerases are attached. That's what this little yellow ball is. They're physically attached. They can't really move away from each other. So we have to move our DNA up to where the leading strand is. So that's why you kind of have that bent position. And when we move on, we're going to go until we hit this primer. So this DNA poly 3, as shown in the second picture, you move until you hit the primer. Once you hit the primer, DNA poly 3 job is done. And so DNA poly 3 is removed and moved to the new spot over here. So you can see it's no longer hanging out on our completed Okazaki fragment. What will happen is that a different polymerase, poly 1, our model polymerase, will come in. It's going to remove the RNA, as we said on our very first slide today, and it's going to fill in the rest of our Okazaki fragment. While the lagging strand polymerase 3 will start to work on our new Okazaki fragment that's right here, right? And it'll start to move down as being shown here. So that is what is happening in general in the replisome. So any questions about that, just off the top of your head? All right. Now let's talk about some of the parts of the replisome in depth. And connected to our polymerases is our sliding clamp protein, which is represented by this little, I guess it's a donut. I was going to say frisbee, but it's a donut. And here's the actual protein. And humans, when we look at humans, our sliding clamp looks like 100% identical to the E. coli one, same shape and everything. So this is called the beta subunit or beta clamp. That's the actual name for it. And it's forming this ring. And so the DNA is going right through the donut hole. And it's attached to DNA poly 3. So this is why poly 3 has such high processivity. If you remember, processivity is your ability to hang on to the DNA. Poly 3 more or less has unlimited processivity because it's attached to a protein that encircles the DNA. So not only does it hold poly 3 there, but it also speeds up synthesis in general. One, because it can't fall off, and two, it's moving poly 3 throughout. It's pushing it forward. Now, when we look at this, even though DNA is going through our beta clamp, we actually see that the beta clamp doesn't really interact with the DNA. Why is that important that our beta clamp doesn't interact with the DNA? So we can move along the template. Yeah, exactly, right? All of this thing, it's a slide. It's just going over the DNA. If it interacts with the DNA, you'll just grab onto it and stay in one place, which defeats the purpose of this whole machinery. So it needs to continue to move. So think of the DNA as like a rail that it's just onto, and like the beta subunit's like a cart on a, cart on a track, more or less. Just moving along on its merry way, bringing DNA polymerase with it. Now let's actually look at a more in-depth view of the replisome. And this whole machinery in E. coli can do about 1,000 nucleotides in a second. So it's pretty fast. As we said, for the polymerase on the leading strand, once it's on there for good. It doesn't need to be replaced. However, when you're doing the lagging strand, here we started at the RNA primer. And so let me get my pen back out. In this form, we are going this way and synthesizing new DNA on our lagging strand. Once we hit our RNA primer, we stop and we need to be replaced. And then we need to reload at our new primer to start again. And so we need machinery to place this beta clamp, because it's the beta clamp that is driving this process and making our polymerase attached to the DNA. So this machinery, as shown here and in this nice little cartoon, has a clamp loader. What the clamp loader does is that it'll take your beta clamp and open it up, put it around the DNA, and then like close it back. So now it's around our DNA. A closed beta clamp naturally will recruit a polymerase. A polymerase really wants to bind to this. Once it's bound, then you can start replicating DNA. So you need to do that all the time on the Okazaki fragment, right? Put new beta clamp, remove old one, put new beta clamp, remove the old one. Now, for the lagging strand, like I said, you need to remove this RNA primer. So what happens when DNA poly 1 hits this primer? Poly 1 falls off. However, the beta clamp will still be there. So you'll just have a beta clamp waiting at the RNA primer by itself. once you have this situation, DNA poly 3, or sorry, I got my numbers mixed up. This is DNA poly 3, that falls off. DNA poly 1 is the one that is then recruited to this beta clamp waiting at our RNA template to remove the RNA template and put DNA there. But that's the process of putting new beta clamps. And looking at this, you can see how I said they're connected. They're connected through this clamp loader complex. So you can see they're connected to the helicase, which is connected to the leading strand polymerase and also the lagging strand polymerase. Here is our DnaG, these little mini bubbles. You can see that. They are going to be adding our new primers. And here's our helicase that is moving in this direction, our DnaB that will continue to unzip our DNA. And further down here would be a gyrase that's undoing all that torsion. So any questions looking at the polymerase machinery here? All right. So this is what I just talked about, but when poly3 hits that RNA of the previous Okazaki fragment, it falls off. The beta clamp stays there. Polymerase 1 comes in, binds to that beta clamp. You remove the RNA, and then you will put in new DNA. And you'll connect the new DNA together. So all of the RNA will go away, it gets chopped up. You put new DNA where it used to be, and then you'll still have a little gap from where the old DNA was to your new DNA. So you just need to seal it back together, right? Because the only time you are making that phosphate bond in the DNA is when you're actively adding a new nucleotide. However, in the Okazaki fragments, every time you finish with one, you're going to be missing that phosphate bond because you aren't adding a new nucleotide. Well, so like this was all RNA, and this A represents DNA, right? Well, I said this was like TCG. Well, I guess that wouldn't be T. Now would it be UCG? So we replace all that. So now it's like A, T, C, G. Since you're adding new nucleotides here, these will all have correct phosphates. This A will have a phosphate to everything to the left of it, but you're not going to have a phosphate here naturally. So you just have to put one in. That's what it's saying here, and seal the nick. Remember, when you have a gap of a phosphate bond between bases, that's called a nick. And we just have to seal that up. So we just put a new phosphate right there. And now you have a complete DNA. So that's what happens at the end of every Okazaki fragment. Let me just see here. Okay. So how do we actually stop this machinery from running in E. coli? And E. coli have this neat little protein pair called TUS and TER. So if we're looking at circular genome of E. coli, there we can see the ORI-C. That's our origin. And there are 10 sites on the E. coli genome that are basically identical, and they're 23 base pair termination sites. So that's why they're called TER. And the way this image is presented, we're starting from each direction here, right? We have the replisome going in each direction because remember, we load 2 DnaBs up at the same time at the origin, and they each go a different way. And if we are going, what did I say, clockwise? So we're going counterclockwise. So we're going this way. We'll ignore the first 5 TER sites. But once we get a TER site that's like open, like here, we will stop. And if you're going the other way, you'll stop here at C on the 6th termination site. And that's what's being shown here in this image. And the left image, it's like we're going counterclockwise and we hit TER J. And if we hit it from one direction, the TUS-TER complex falls off and we can continue. However, if we go and run into like TER A, we're coming at it from the other direction. And what happens is this TUS-TER will flip out some nucleotides and make this replisome just stop dead in its tracks. Because since these nucleotides are no longer facing the right way, our DnaB can't work anymore. It has no nucleotide to bind to, so it can't continue to unzip. Now, when looking at the system, these termination sites, so the termination site is the DNA portion. It needs a protein called TUS. That's why it's called TUS-TER. The blue here is the TUS protein. And it'll only stop if the TUS is there. So if your organism has no TUS proteins, no matter what, your replisome will never stop at a TER site. It'll just keep blowing past them. But that actually doesn't matter. Because let's imagine you have no TER sites. You're going in each direction with replisome. Once they hit each other, they're naturally going to fall off and replication will stop. So it's kind of weird that these TER sites exist to stop our replisomes because there's already a system in place to stop the replisomes when they crash into each other. But when we look at this, we see this TER-TUS system conserved, right? It doesn't go away. It's in organisms with circular DNA. This means that having this system for whatever reason is some kind of advantage. We might not know what it is, but just because it's there and doesn't go away when we look at different species means it probably is doing something helpful to give them an advantage against organisms that don't have this system. But yeah, that is how you stop replication in E. coli. You either hit a termination site and then the other replisome will come and hit that, the replisome that's stalled, or they just hit each other. So questions about termination. All right, so one more slide here. And that just talks about fidelity. Fidelity is the ability to replicate DNA without making a mistake. And so everything we looked at here, all our machinery basically can replicate DNA perfectly or near perfectly. Like I said at the very beginning of class, you have one mistake every 100 million to 10 million base pairs. So the number I gave you before, 1 million to 100 million, that was just for the polymerase by itself. Once you have the whole replisome, it gets even better at not making a mistake. You're looking at 100 million to 10 billion base pairs before you have one mistake. Why is it so good? One, you have balanced levels of NTPs. So if you had like a lot of like ATP versus the other NTPs, you would probably add in A's on error more often. But since the cell is going to keep roughly the same levels of all these nucleotide triphosphates, you're less likely to make an error because of that. Another reason we have fidelity is because adding a new nucleotide is 2 steps. Remember the open and close conformations. So the first step is just binding in the open form and seeing if you have the correct hydrogen bonds. If the Watson-Crick hydrogen bonds are not present, you're not going to be moving to the closed condition. You're just going to stay there in the open, and that nucleotide will just move out of the pocket, and a new nucleotide will bind. If you have the correct Watson-Crick base pairs there, then you go in the closed. So that open and closed mechanism is one safety check to make sure we aren't adding nucleotides in the wrong order. But another safety check onto that is if we do add the wrong nucleotide, we can actually use our exonuclease and remove that. So if an error is made, we can just cut it out as soon as we make it. And on top of that, let's say an error is made, you have a bunch of enzymes. E. coli does, humans do, everything does. That will just scour the DNA for any types of damage, be it chemical, synthesis, whatever, and fix that damage. Which means that although organisms mutate, they mutate extremely slow because we're really good at preventing mutations. And that's why when you talk about evolution, you're not talking about hundreds of years, really. You're talking about millions of years to get large scale change because it's very, 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 very gradual. Now, when looking at where error happens, it usually happens adding the first few nucleotides. That's why we have this RNA primer, right? We just have DnaG, just add RNA. And who cares if it makes a mistake? All that RNA is there for is to provide a phosphate, an open 3' phosphate for our poly 3 to work with. And then once that 3' phosphate is present, we start adding nucleotides to our primer. That's why we chop off that primer at the end. And that's why we use RNA. Because if you're going to make an error, it's going to mean the first few nucleotides. So why not just make it with disposable nucleotides that you don't care that are there anyways, and you can remove them. So all of that combined, make sure that our DNA is replicated with high fidelity. And that is the end of everything I had to cover for today. So any last questions people might have? And if you have those, feel free to tell them out. Otherwise, just as a reminder, in case you came in late, test 3, next week, Tuesday. These slides were test 4. They should be in the test 4 folder. So this whole PowerPoint. Homework. So there will be homework for this. But it's due next week, Thursday. Since the material we covered today is not going to be on test 3, I don't see any reason why you should rush to finish it before you take test 3. So this will be due next week, Thursday, which means on Thursday next week, you will have 2 parts, part 1 and part 2 for homework. So be on the lookout for that. Lastly, no live lecture next week, Thursday. I will not be around. So I will put up a video lecture sometime before then. Other than that, that's all I have for you. So if you have any questions, as always, don't hesitate to let me know. Otherwise, thanks for coming. Thanks for watching on YouTube. And I will see everybody later. Have a good rest of your day, everybody.